Good morning, everyone. I'm happy to moderate this session. I gave a bit of introduction the other day, but a little more is I'm a veterinarian, attending veterinarian, and I'm retired IO. That's the gray hair. I had 25 IACUCs or ethical review bodies reporting to me at one point, so that kept me busy. But today I'm real excited to do the study animals, a nice introduction. I am a human. If you want to study me, you need an I or B. So we're not covering that today. So we'll go into the other species. First off, I would like to bring up Christopher Parkinson. We're going to go over reptiles and snakes and amphibians. He's a professor of biological sciences at the Joint Appointment in Forestry and Environmental Conservation at Clemson University.
All right, welcome. So I don't stand behind a podium. It's not who I am. Because I do things like that. So Bob asked me to join this to talk to you about reptiles and amphibians and go through that. And so the way Bob and I envisioned this symposium, this piece of it, is really a feel-good session showing you the diversity of what we have to offer. And so talking through it. And so most of you probably have faculty that do research on reptiles and amphibians. And it's getting larger and larger in terms of research on those groups. And so if we look at reptiles, consists of alligators, crocodiles, lizards, snakes, turtles, tuataras, and these weird little things called amphisbaenids. Most of you probably never seen or heard of them, but here's a picture of Rhinura floridana. It's found in Florida mainly. It's the only amphisbaenid found there. And you have, 10,450 odd species of lizards and snakes, 300 species of turtles, 23 species of crocs. How many of you have researchers that work on crocodilians at your institution? So it's getting larger. There's a lot of that. And so think about what would happen if out of the blue a faculty member walked into your IACUC with a protocol to work on crocodiles. How is your IACUC going to react? Amphibians, almost 8,000 species of amphibians. So this broad group is amazing in terms of biodiversity. And we are lucky enough that we can study a lot of these in captivity. If you don't like snakes, you might as well shut your eyes. I'll just tell you that now. My mother has never forgiven me for going to work in this field. But the diversity of snakes is amazing. Feeding ecology, you have snakes that specifically feed on eggs, Dacybeltis from Africa. Or you have pit vipers that are we just described this species from Costa Rica 2 years ago. Or king cobras that feed on snakes. How do you deal with that? How does your IACUC deal with, I got to come up with snakes to feed my snake? These are questions that I have to address with my research program. And you will have to address. And I have just experienced what I've had a lot of people experience in that I moved institutions after 17 years at the University of Central Florida and training that IACUC and that EHS group and things like that. I've moved to a new institution, Clemson. Had a ball. It's been fantastic. But I forgot about the reaction. Oh my God, he works with snakes. Oh my God, he works with venomous snakes, which is my area of expertise. It's been really interesting. But the diversity is amazing. From the federally endangered ridge-nosed rattlesnake here, you can't really see it, which is the coolest thing, but there's a 2-meter boa right here that James Adam took a picture of in Honduras. Lizards, absolutely amazing diversity. Here's our horny toad from Texas, southeast. But look at Moloch from Australia. Very similar. Can somebody tell me, other than Michael, what these guys eat? Ants. Let's think about that one. How are we going to get an ant colony to feed our, okay? And then you have Todd over here working with Nile monitors in South Florida. If you want to see weird reptiles and amphibians, go to South Florida. Okay, my daughter just caught this chameleon down in South Miami. Okay, beautiful, great population of 3 species of chameleons that live in South Florida. But a lot of these are used in research. They're kept in captivity and used in research. Anoles, the little guy right here, very common species that we have brown anoles, green anoles, and I actually use anoles for LD50 work in snake venom. It's more of a prey item that we utilize. But let's think about dealing with the IACUC issues of that. Okay, so the diversity is amazing. This mole skink right here, we have a U.S. Fish and Wildlife program working on mole skinks, one of my graduate students. They thought it was very rare and everything, but nobody actually had really went and looked for it. They're semi-fossorial, they live in the sand. You have to hit them at the right temperature to find them, and then you find lots of them. You just have to know where to look. So bringing that expertise in to think about these aspects. Crocodilians and turtles. How many of you want to have researchers that deal with large crocodiles like that? I mean, I've done it. My EHS, they don't like me sometimes. But it's all about training and educating because it can be done safely. It really can be done safely. Or tortoises. Okay, we program over at Kennedy Space Center coming up with ways to deal with tortoises going under railroad tracks, because if they get caught in the middle of tracks, they die pretty much. They dehydrate and die. One of my graduate students was able to grab this. It's a natural history note. Didn't think alligators actually ate tortoises. It actually happens more frequently when one of our radio telemetry kept coming up in the pond. And we couldn't figure out why. We did later. Okay, marine turtles. We'll talk about marine turtles here in a second. FAU is represented here, and I'm actually going to show you some pictures from there. But a lot of programs deal with marine turtles, and the layers that you have to deal with are pretty amazing. Frogs. Absolutely cute and fun, but there's an amazing amount of things we have to think about when we bring frogs into captivity. A lot of researchers are working on dendrobatids, poison arrow frogs, but Phyllobates, this guy right here, Terribilis, produces toxins that will kill you. These are poisonous. They're not venomous, they're poisonous. Okay? Natives utilize Phyllobates on arrow tips for hunting. Think about that neurotoxicity. Okay? But these are used a lot in research. How about eggs? Should we cover those eggs as an IACUC? We can have that conversation. Or right here, we're in the midst of discussing whether we should use gloves when we're out in the field handling. Yesterday in our little breakout session, we got into a discussion between some people on gloves and what the opinion should be. For sure, with these rhonids, which are an endangered species found in Arizona because of chytrid, wearing gloves is highly, highly important. Okay? But the diversity is amazing when it comes to reptiles and amphibians. Salamanders. It's been great moving to Clemson because now there's salamanders in Appalachia. And it's fun to go out and flip and look for them and things and take my daughter out hiking. But salamanders are common. We have a researcher at Clemson who has a whole room full of salamanders, and it's a cold room, okay, to keep the temperatures appropriate. But these are things that are easily dealt with in captivity if you have the expertise. And if you don't, you make a phone call or an email to help bring in somebody who does to help facilitate that IACUC aspect. So areas of research. University of Kentucky has an axolotl colony. Huge axolotl colony for ambystoma genomics and genetics that you can go and utilize. Evolutionary biology, anywhere from ecomorphology, ecology, conservation, disease ecology now. So there's a lot of aspects about it. And last, with venomous snakes, there's a whole medical drug discovery aspect. My area of research is venom evolution and venom toxicity, and we use genomics to look at the evolution of venom. But we have to have the animals to do that. And we have to work with those animals in captivity or in the field to do that work. So that adds a whole layer of EHS to it. Matter of fact, I have an EHS meeting on Friday that just got scheduled today because of my AUP was going through, and the director of EHS wants to chat with me about it and the 27 SOPs that go along with it. So these are things that we can solve with communication. How do we deal with snakes in captivity? Michael and I were talking about this yesterday. Housing venomous snakes in captivity. We can do it. It's actually very easy. You have multiple doors that go through and keys. All these cages have locks on them. They're slip-out cages that have wire tops on them. At the University of Central Florida, one of the nice things when they designed one of the animal facilities for me, they have a push-button automatic 911 telephone right there. You step out of my room, and you hit that button, and you're automatically on the phone with police and fire. What do? I bring in police, EMS into my room, go through with them every 6 months. They know what I'm housing, how to deal with it, and work with it. It's setting up that communication relationship before those animals are even in the area. Lizards. Jonathan Losos maintains or used to maintain a large colony of anoles. Rat cages work great for this. The large ones, you can see them right here, lights on top, feed them Drosophila and crickets and things like that. They work very well maintained in your vivariums. Marine turtles. How many of you have people or researchers that work with marine turtles? Okay, a lot of us do. How many of you have them that actually work with them in captivity? Okay, a couple. So this is one of the FAU groups. And what's amazing is this. So they work with a lot of juveniles, and one of the things you probably can't really see is this individual is tethered. They have a tether system so it doesn't hit the sides of the cages. They're always swimming, but it's a safety feature. We have to think through those processes. Anna Savage at UCF works on basically ronid frogs. They carry a lot of BD, okay, chytrid. And how do you deal with it? Mouse cages work great. So thank you, mammalogists. You've helped us, okay? But setting up these facilities in-house works very well. But there's this diversity of reptiles and amphibians that is used in research. And we can apply a lot of things to that to build your research program and facilitate your researchers. So here's one of the hard ones that we have to deal with. Non-traditional food items. I mentioned it jokingly with king cobras, but think about these sorts of things. For most snakes, we feed mammals. And so I had at UCF an MOU with our IACUC that all the culled mice and rats were utilized for us. Okay? So anything that did not have any experimental work done on it, that was a cull, was frozen in packs of 10, and they went to the Parkinson snake freezer, and that's what we utilized to feed. And so it worked very well, the 3 R's. Okay? But you can see this little Nasudum, this little rainforest viper here. It's a lizard, loves to eat lizards. Think about that. Okay? Eggs, crickets, earthworms, bringing those things in. I've had, as the chair of the HERP, ICS, and HERPS committee, I've had vets call me and say, you know, how do we deal with bringing these odd food items into the vivarium? What are the issues with that and things? There's a huge herp community out there that have specialist groups that raise mice and rats to feed reptiles. You can call up Rodent Pro and get 200 frozen mice sent to you via UPS. I don't want those mice in my vivarium, okay, because of zoonosis. But we have to work with our groups to figure out how we deal with this. All right. So these are things we have to talk about. One of the things, daily, weekly, monthly feeding schedule. It's dealing with reptiles and amphibians have different feeding schedules. Amphibians, a lot of times you need to have fruit flies in there a lot for them, like shrews almost. They're constantly feeding on. But snakes, when I tell them that they might be fed once a month, I got a little nervous about that. Or when they don't feed for 3 or 4 times. Well, there's a problem. We have to deal with it. No, there's not a problem. All of a sudden, they go back on feed. And so these are things that you need to talk to those experts that understand the husbandry and care behind them. Daily check. With mammals, you need to look at them every day. With a lot of snakes and things like that, you're actually causing issues if you go in and disturb them every day. Okay, filling water bowls, doing things like that. So we try to go in every day, but it might be every other day depending on what animals we have in captivity, what we have in the collection at a time. I've got a huge number of people to thank for this, but just so you know, you have resources out there on reptiles and amphibians. I'm simple, viper@clemson.edu. Thank you. Do you want the podium?
Thank you. Okay. Let me just. Stay tuned. Give you a quick introduction. Our next week, I guess evolutionary. We're going from lizards to birds. We're flying now. So I'd like to introduce the next speaker, Ellen Paul. She represents executive director of the. That's. I don't know. It wasn't.
Does anyone know how to take this off of auto?
Okay, hold on.
This is normally where I call my husband and say, honey, help. He's my tech guy.
It's not
It's not running now. Okay.
now.
Okay. But when you, but now it's not on. Oh, it is on the screen. Okay, good. We're good.
All right.
Okay.
Well,
It seems to be okay now.
well, it's not on
Oh.
screen, so.
It is. It is.
Well,
but you
no,
got that.
I got.
Okay. Take it off of here, please.
Or you want to.
I don't use a PC, so I don't speak PC. I speak Mac. Sorry, I speak Mac, not PC, so I don't touch PCs because I'll screw it up. For this reason, right? Thank you. I can introduce myself.
Okay.
Okay, thanks.
Here's someone going to talk about birds.
Thank you.
She knows a lot of stuff.
I don't know anything about PowerPoint on a PC,
And
but
they're not
birds
as
I can
dangerous as
do.
the previous guy's stuff.
Oh, you're going to find out. Okay, so I am Ellen Paul. I'm with the Ornithological Council, which is a consortium of 11 scientific societies of ornithologists scattered throughout the Western Hemisphere, which makes us sound like we're really big, but it's actually just me and a huge board. So I report to 22 bosses. Really interesting dynamic. Even though the societies are in the Western Hemisphere, in fact, they have members from all over the world, and their members work all over the world. So in practice, it's actually international. And I actually have just changed my take-home message for you, so pay attention. Based on the comments and questions and private discussions that I've had just in the past 24 hours, it's clear to me that some of you haven't heard of us, and even if you have, You aren't using us as a resource. If you have questions about permits, which is a big issue with birds, or about ornithological research generally, please call us. You will not get a biased answer. We do not always support everything our members want to do. And if we don't have the information available, we will find experts for you who do. So with that being said, I am going to channel my best David Attenborough here. Birds are spectacularly beautiful. They are just fascinating creatures of the air. I spent hours trying to pick which ones because there are just so many. Watch this. You may have seen this on TV last year. That is a superb bird of paradise doing its mating dance. I mean, with all due respect to the herps, you got nothing on us. How insane is that? Okay. What IOs, the members need to know about ornithological research. First off, what laws do and don't apply and how, the diversity of birds, what we study, how to minimize impact, care in captivity, and avian zoonotics and other occupational safety questions. Going to cover that all fast, so fasten your seatbelts. How many bird species? 10,534, give or take. If you're a birder, you hope for what's called splits, meaning that if you've seen a bird and they now split it, you get an extra one for free without having to go back to that country. We call it an armchair tick. They are in 40 orders, 240 families, and 2,276 genera. Size extremes, 185-pound bird, a bird that weighs just a couple of grams. It's really amazing. Most fly, as I'm sure you know. Some don't. This is a kiwi. It just sort of toodles along beaches and in the forests, foraging under kelp and under moss. Some swim. Some take Uber. That is not photoshopped. That's for real. Some fly coach. These are falconry birds in Saudi Arabia. It's a very big thing in the Middle East, and the sheikhs have planes that are specifically dedicated to transporting their birds around the world. How they eat. Most birds, even though they have a preferred food item, are opportunists and will investigate and eat whatever they can find, which is not always a good thing because sometimes it turns out to be toxic. French fries, a big problem, a very big problem. You have California condors hanging out at McDonald's, looking through the garbage in the parking lots and in the trash cans. These are not condors. Vampire birds, very, very varied diet. I mean, this is really a balanced diet. They actually eat the parasites that are on giraffes and other mostly ruminants, and they also eat the dander. They also will peck blood, peck small holes and drink the blood. It's kind of gross, actually, but they'll eat whatever they can get from those mammals. We have nectivores, of course, birds that have all sorts of specialized adaptations. So it isn't just a question of providing nectar. It's a question of providing it in a manner that actually allows the birds to eat it. We have insectivores. These are bee eaters. Obviously, that could be a little bit difficult to keep in captivity. They don't just normally sit on branches and wait for the insects to come to them. They normally dart out just as swallows do. We have carnivores. Sometimes my critters eat Chris's critters, evening the score. 2 out of the 3 of those, we do okay. That's a secretary bird from Africa. Sometimes they only eat dead food items. So eagles, for example, they eat carrion. We have fish-eating birds. And we actually also have bird-eating fish. I'm sure some of you have seen the meme that's going around, or the article rather, that's going around the internet this past week, where fish have been leaping out of the water and catching birds out of the air. Not fair. So what do we study? We study everything: taxonomy, anatomy, physiology, energetics, diet, foraging, every aspect of mating and breeding behavior, predation and predators, both sides of the coin, ecology and behavioral ecology, flight and other forms of locomotion, migration, genetics, population genetics and population structure, vocalizations, learning and social behavior, disease ecology, and zoonotics. Some of those cross over to directly benefiting humans, obviously in the case of things like highly pathogenic avian influenza. Ecotoxicology can also benefit the human population, if only we would listen. Canaries in the coal mine. And then any number of conservation challenges. I have just a few examples here: telecommunications towers, window glass, power lines, free roaming pet cats and feral cats, non-native species of plants, habitat loss, and habitat alteration. So I've tried to give you a broad brush here. There are many others. So the take-home message here is that one size truly just fits one, maybe two or three. If you look at all of the different factors that go into a decision about how to study a bird, whether in the wild or in captivity, it's a huge number of factors. So here's just one example of capture methods, 4 different capture methods for similar kinds of birds. These are basically aquatic birds, right? We have mist nets over water. That's actually a rather large bird to be caught in a mist net. It's really tough to catch large birds in mist nets. They tend to go through them. We have cannon nets and rocket nets, and also swoosh nets. We can also corral them at certain times of year when they aren't very good at flying because they've molted their primaries. And we have funnel traps, and I don't know if you can see that because it's well camouflaged by the vegetation, but these are birds that normally live in dense vegetation, and so if there's a food source in there, they don't recognize that that's a trap. That's something it would normally go into. And this is just 4 out of many, many capture types. In terms of marking, again, a few examples. The little yellow guy on your bottom right is a most typical example. There is an aluminum band that's issued by the USGS Bird Banding Lab. There are also plastic color bands on that bird, and there can be up to 4. You do need special permission for the color bands. You need a permit for banding generally, but special permission for what's called auxiliary markers. We have a bird with a nasal saddle. Only certain ducks can have nasal saddles and nasal discs because they do get caught in nets and underwater vegetation. We have a geolocator on the little guy in the middle. These are extremely lightweight, but their use is somewhat limited. We have a radio transmitter on the bird at the top, which is a problem because of the antenna. You can imagine that you couldn't use those on ground-dwelling birds. We have a patagial tag on the crow. That's really so if you need to identify a bird using your binoculars and without recapturing it, it's really hard to recapture crows. They're really smart. They fall for it once. That's it. We have neck tags or neck collars, which are used primarily in the context of waterfowl management. So again, just a few examples. So let's switch to regulatory. Our birds covered under the Animal Welfare Act, and I'm sure Nicolette's going to fall off her chair when she hears this, but we had the discussion yesterday, so she's prepared. The extent to which birds are regulated is limited. The regulatory exemption was eliminated in 2004 following the statutory change in 2002 that required coverage of rats, mice, and birds except those bred in research. However, there is no, quote, rats, mice, and birds rule. And again, with deference to USDA, Nicolette's exactly right. It's rats of the genus Rattus and mice of the genus Mus, so I've overstated that here. APHIS published an advance notice of public rulemaking in 2004, but never actually published a proposed rule. As of 2017, the entire process appears to be officially dead. In June, OMB published what's called the semi-annual regulatory agenda, and it has 3 lists: regulations that are in some process of being written and formalized, things that are on the inactive list. They're not dead, but they could come back to life sometime. And then also long-term actions, which are things that are not expected to move in the next 12 months. This rule appeared on none of them. So as best we can tell, it's officially dead. However, as I'm sure you know, birds are covered under PHS policy, which covers any live vertebrate. Many ornithological studies likely qualify as field studies under the Animal Welfare Act. However, PHS policy includes any activity that is likely to alter or influence the activity of the study animals or others in the area. However, and this is a huge one, federal grants and contracts policy requires Animal Welfare Act compliance. I don't want to get into all the details. Come to the compliance session tomorrow and I'll explain this in more detail, or find me. But there is a form called Standard Form 424B. There's also one called 424R&R, which does not stand for rest relaxation. It stands for research and related activities. And it says, this is something you have to sign when you get a federal grant or contract from any agency, will comply with the Laboratory Animal Welfare Act of 1966. Period. No exceptions based on whether or not APHIS has actually promulgated a rule. So even though there are no standards currently for inspection purposes, no rats, mice, and birds rule, the registration and protocol review requirements must still be met. And yes, we have been discussing this with APHIS's general counsel and with the deputy director. Permits. Actually, you know, that 424 also has a provision about drug-free workplace, and I know most scientists don't like that one because they have to deal with permits. But in the U.S., permits are needed to study nearly every bird species found in the U.S., which is 1,026 species on this list. It has nothing to do with actual migration. The title is misleading and has to do with a legal issue. Endangered Species Act, 81 are listed in the U.S. as endangered, 18 as threatened. Outside the U.S., 214 and 17 as threatened. The Bald and Golden Eagle Protection Act is obviously just 2 species. Generally, under the Migratory Bird Treaty Act, no permit is needed if not capturing or handling. The law doesn't cover perturbance or harassment, this particular law. So you can see where there's a mismatch there between that and the Animal Welfare Act. For the ESA, however, a permit is needed even to survey. The law does cover harassment in this case, which is defined as an intentional or negligent act or omission which creates the likelihood of injury to wildlife by annoying it to such an extent, go ask them, are you annoyed? To such an extent as to significantly disrupt normal behavioral patterns which include but are not limited to breeding, feeding, or sheltering. Disposition at the end of the study, Animal Welfare Act, Public Health Service, U.S. Principles, and the guide, or those 3 rather, say nothing about what to do at the end of the study if you have taken an animal into captivity. The guide says when they're removed from the wild, the protocol should include plans for either a return to the habitat or their final disposition as appropriate. The problem is that federal and state permits, usually state permits, may require, in some cases, or prohibit, in most cases, release of the animals to the wild. So you have to, the researcher really has to read the permits, and it's not the IACUC's responsibility, but it's the IACUC's responsibility to make sure the researcher knows what the permits say. And I can tell you a fact that they often don't read their permits carefully. So that's something that IACUC really needs to keep on top of. Not enforcing the permit, that's for a different agency, but making sure that the researcher reads the permit carefully and frequently, because they do forget. Euthanasia is another problem where we have a bit of a mismatch. Currently, if a permit includes authority to euthanize a bird, it will almost certainly limit the methods to AVMA approved or approved with conditions. As a result of discussions that we've been having with them recently, they are considering adding or is approved by the IACUC, because clearly that's legitimate. It's allowable under both Animal Welfare Act regs and under PHS policy. Okay, now we're going to get to the serious stuff. Now, admit it. I know some of you are young, so this may not apply, but don't you get just a little creeped out when you see a bunch of birds gathering on a playground all these years later from when you first saw that? Okay. This is what we call one stupid human. That bird is 175 pounds, and it is armed with extremely sharp claws, and they have been known to attack humans. They can do serious damage. That's a cassowary. Talons. All the raptors have these guys. Well, so do parrots for that matter, but with talons they're especially. I have a cockatoo, which is why certain people are laughing. This is really scary. And notice, no gloves. Okay. Daggers. Loons and certain other water birds have extremely sharp, strong bills, and they will attack, and in fact, they often go for the eyes. So even though I joke about the lab standards being inapplicable to wildlife research, this is one case where goggles are actually appropriate. They have gone after eyes, they will. How birds know the most sensitive place to go after you, I don't know, but ask anybody who owns a parrot right here. Birds can be toxic. How many of you knew that? Yay. There are 5 species of birds, 3 of them are called pitahuis, which is such a cool name, that actually excrete a toxin in their skin, and of course it gets onto their feathers. One case where I would say, yes, it is the thing that the IACUC has a reasonable right to say, wear gloves. Normally I would not say that, but in this case I would. Zoonotics, really in terms of encounter rates, the major threats are vector-borne diseases that you would encounter in the field, whatever your reason for being outside, Lyme, Hanta, and so on. Pathogens such as toxo and vibrio, those aren't unique to birds or ornithological research. The ones that are commonly encountered in terms of birds would be West Nile. Birds are not competent vectors, and this is more a question of avoiding mosquito bites. Highly pathogenic avian influenza, the risk depends on the strain. I won't get into more detail there. So here's how they used to sample ducks for avian influenza back when it first was a really big, serious concern. Gloves, gowns, you can't see the goggles in there, but they're wearing goggles. This is gloves and goggles, and this is what it looks like today. I don't know how well you can see the guy holding the oyster catcher. No gloves. Okay. Now the sparrow, they're normally highly pathogenic avian influenza is more of a problem with waterfowl, but the fact of the matter is that ornithologists almost never wear gloves, even where there's an active outbreak. Raptors, there's a lot of raptor people out there who do not wear gloves. And you can see there's a wide range. And you'll also see a duck person at the bottom. Again, not wearing gloves. And we're not dying. You know, this is kind of like when we were kids and we all rode in our parents' cars without seatbelts. We're not dying, guys. This would be my least concern. Studying wild birds in captivity gets incredibly complicated in a hurry for all the reasons I've said. Obviously, there's quarantine that is mandatory, not only for the sake of the individual birds, but any other birds that you might be introducing it to. There's an appropriate enclosure issue, allowing flight or other forms of locomotion. Social behaviors in the wild, such as group housing or preventing aggression and mating, are key. Appropriate diet and foraging opportunities and feeding methods, really tough to do for most species, especially the insectivores. And enrichment for some species is absolutely critical. You got to provide these guys with their own laptops. I would like to leave you with one example, and this is drones or small unarmed. So sometimes they are armed. Small unmanned vehicle, autonomous vehicles, drones. They're only been in use really for wildlife since 2006, and I'm just in the process of completing a literature review that will be an addendum to our guidelines for the use of wild birds in research. And it's amazing to me how much variation there is, and what we know so far is just really complex, just since 2006. And I only did birds. So there is a question of fixed wing versus rotary, and if fixed wing, what shape? The distance at launch from the birds you're going to study, the altitude, the change in altitude, and the angle of approach, straight line transects versus circling versus hovering, speed, surveys versus nest inspections, what species you're talking about, the presence of predators, training, and the condition of aircraft. So you can see there's no way to have a rule or even a suite of rules. The rule is consider these things. The rule is go out and read the literature. The rule is go out and talk to people who have been doing it. The bottom line is the impacts of any method for any study are highly variable. The impacts are not yet well studied, at least in the case of drones, and the impacts seem to be far less, interestingly, than those resulting from foot approach or boat approach. So the standard isn't no impact, the standard is less impact. And so drones may actually be a good thing in that regard. Resources generally, there's on your thumb drive, there's a whole slew of resources, but the ones that I'd like to highlight to you here are guidelines to the use of wild birds in research. It is peer-reviewed. We do update it periodically. The Model Wildlife Protocol, ours does vary somewhat from the one issued by the mammologist, primarily because we've added a section about when PHS policy applies, because it's not just PHS funding that triggers PHS compliance. And avian zoonotic disease, we have a peer-reviewed fact sheet. There was also an OLA webinar that I gave in March on permits, and it pretty much is everything you need to know about permits. And it is archived on the website, on OLA's website. And then birdnet.org, we're just about to launch a new website where things will be much easier to find. For keeping birds in captivity, our guidelines do talk about it to some extent. The American Zoo and Aquarium Association has taxon advisory group animal care manuals, and people in the zoos are very good about sharing their knowledge. So talk to zookeepers, talk to others who have kept birds in captivity. And that's it.
Thank you. Very good. Although I think steaks still scare me more, but if I'm walking out of McDonald's with a French fries, I see a California condor staring at me. He's getting my fries. This one? So how do you like to start? Okay. Person who's already been introduced, Robert Sykes.
Okay, yes, I'm wearing the ALAC tie. I will take it off after the ALAC session this afternoon. I would have waited and put it on after the ALAC session, but the fact is I wear them so seldom they're hard for me to tie. So you put it on once and forget about it. All right, I get to talk about mammals, the least speciose of all of these groups, the least impressive in terms of colors and all of that, but the ones that you're probably most familiar with. I mean, most of you are going to have rodents, at least in your facility, most of you. So everyone's familiar with mammals. There shouldn't be a lot to say here, right? Right. There are three major lineages of mammals. If you go back and look at the diversity of mammals, the radiation, the first lineage are these guys. You're probably not nearly as familiar with these. These are called prototherians. They're the mammals that lay eggs. They are the most primitive of the living mammals. So you've got an echidna on the top left there. You've got a duckbill platypus on the bottom right. Mammals that lay eggs. Mammals that live on arthropods. Mammals that some authorities will say really aren't mammals. Some authorities will say that these are actually living therapsids. What's a therapsid? Most of you probably had this animal in your dinosaur kit when you were a child, right? It's not a dinosaur. This is a therapsid. This is the lineage of animals that gave rise to the mammals. And if you look at those prototherians, they share a lot of characteristics with these guys, including the egg laying. So bringing them into captivity or studying them is a very, very different creature than studying these mammals that we're much more familiar with. Lots of physiological differences in addition to anatomical differences. These prototherians, their thermostat is set way low compared to us. We typically worry about rabies with mammals. Nah, not with prototherians. Their body temperature is set too low. The rabies virus wouldn't survive. The second lineage of mammals, much more closely related to us, the metatherians. They don't lay eggs. So an example from North America is our common opossum on the top right there. And the coenolestid, bottom left, this is a little Central American, South American marsupial, if you will, a metatherian. They don't lay eggs, but they do give birth to embryos. Maximum period of gestation for a metatherian, 28 days. Some of these, their entire gestation lasts 7 days. That's it. Then they give birth to an embryo. The embryo goes through the rest of the development in the mother's pouch or attached to a teat. And that's their nutrition. That's their umbilical cord. So when do you start calling it an individual? The ones that you're much more familiar with, the eutherian mammals. These are placental mammals. This one is a grasshopper mouse, one of the species of grasshopper mouse. Okay, you've got these in captivity. You're all familiar with these. These are just like us. Keeping a rodent, you know, if you can keep a rodent, you can keep any rodent, right? No. This is a rodent that's a carnivore, almost a pure carnivore. 90% of their diet in nature is animal matter. That's scorpions, that's grasshoppers, that's bombardier beetles. This is a guy that will grab bombardier beetles. You know what bombardier beetles are, right? These little beetles that squirt formic acid out of their abdomen, so they're really nasty. That's their defense. These guys learn how to do it at a very young age. They'll grab the bombardier beetle, cram its butt down in the ground, and eat it from the head down. Scorpions, same way. They'll learn to take the tip off of that scorpion in a heartbeat. They don't get stung. And turns out that the scorpion venom doesn't bother them. We've got rodents that, in the bottom left, live completely without free water. This is a kangaroo rat, banner-tail kangaroo rat. Bring these into captivity. How many of you have K-rats in captivity? Not a single hand? Oh. Okay, have you had the VMOs come through and cite you for not having water bottles on the cages? They live completely without free water. Keep them in captivity 2 years. Never a drink of water. In fact, if you provide them free water, you change their physiology enough that they become addicted to water. They get by without free water because they have the longest loop of Henle, the functional unit of the kidney, of any known mammal. Live completely without free water. If we could domesticate these things, bring them into the lab for a lab animal, a lab model, fantastic. All of your animal care people would love it. Change their bedding once a month. Never hang a water bottle. Their feces is very, very dry. They're recovering all of that moisture. Flip side of that is the animal in the upper right. This is an animal that lives in free water. That's the world's largest rodent, the capybara. Substantial difference in kidney function there, obviously. Back to these grasshopper mice. This is a different species. This is one near and dear to my heart. This is a northern grasshopper mouse. Anybody have these in captivity? Swing and a miss. A desert-adapted animal. Bring them into the lab. You better have the substrate right. Look at the substrate under the animal on the top left. Does that look like Santa chips? It's a hardwood bedding that's really fine ground. These are desert-adapted animals. They produce a lot of oils through the glands, and if you keep them on Santa chips, they turn into grease balls really, really quickly. You need a really fine bedding so that they can sand groom. It's just a replacement for sand for them. Keep them on the right bedding, they stay looking nice. Put them on Santa chips, grease balls almost instantly. So here, this one on the right here is actually eating a cricket. So had these in captivity for quite a long period of time. In fact, we were interested in these animals, besides behavioral aspects, for their communication. We think of quiet as a mouse. These rodents don't vocalize, or at least they squeak every once in a while, right? Not this one. This predator behaves like a wolf in more ways than one. This little 30-gram animal, the males especially, will stand up on their back legs and howl. It is so high-pitched that you can barely pick it up. It's at about 20 kilohertz. So it's really at the top end of human hearing. So I had a graduate student that really wanted to look at these vocalizations because, and it turns out that this howl is really a defensive thing. They're very territorial, and this is how they're establishing their territories, saying, this is mine, just like a wolf howl. So we set them up for, we built a sound chamber in the lab, put these animals in to record these vocalizations. And we thought that the overtones or the harmonics in those howls would be important in determining how far away the animal was. So we had microphones that would go at really high frequencies. And we had a serious problem. We ran into a roadblock, actually, because when we had the animal in the chamber, it obviously was not vocalizing. We were getting noise in our sonograms up at about 50 kilohertz, so twice as high as humans can hear. This was in an adjacent satellite building. We went through the building and turned off every piece of electronic equipment. We unplugged freezers. We unplugged refrigerators. If it was electronic, we killed it. And the harmonics were still coming across. They were intermittent. This noise was coming across. We could not identify it. But it wasn't there when the mice were not in the cage. Turns out it was the mice. Who knew? Now, when you fill out your ALAC program description, you talk in there about keeping noise down in your environment, right? You talk about vibrations in your environment, right? How do you test that noise? How many of you test it with microphones that go up to 100 kilohertz? Nobody? That's their environment. We don't have a clue what sort of noise we're putting in these animals' environment. And the 50 kilohertz band is where these animals do their close communication. You don't get that when it's one animal. You get that when there's multiple animals in there. So by having this high frequency noise that we have no clue is even in our facilities, we're actually disturbing those animals in terms of their normal communication. We view the world through our lenses. not through the lenses of the animals, and theirs is what matters. Remember, one of the differences between biomedical research and wildlife research is we're doing this to understand the animal, to understand how they fit in their environment. We don't want to alter that unnecessarily. And sometimes we don't know that we're altering it. So these animals live in an acoustic, a very important acoustic environment, in part. They also live in an environment that's an olfactory world. These animals, like many other rodents, have ventral scent glands. If you leave a grasshopper mouse, most mice, in their cage long enough, don't change the water bottle, don't change the sipper tube, don't change the hopper feeder. After a little while, particularly with grasshopper mice, you notice there's a gray scum, a film around the end of the sipper tube. And then it appears on the bars of the feeding hopper. It's because they're marking it with their ventral scent glands. That's their water bottle. That's their food. That's their environment. When we come in and change that environment twice a week and give them fresh cage, fresh bedding, fresh water bottle, fresh sipper tubes, fresh everything, put that in perspective. Everyone have a good sleep last night? Think if the hotel staff had come in at 2 a.m., grabbed you out of bed, put you in a new room. No, you cannot take any of your belongings. Here, you live here. And then we're going to do that again 3 days later. That's what we do to a lot of our animals. What works best for these guys? If you want them to breed in captivity, they've got to be comfortable in captivity, comfortable quarters. That involves changing the cage and bedding on a much slower schedule, like 10 days to 2 weeks. And when you change it, never change everything at once. Always leave some component behind so that something is familiar territory. Is this standard practice at your facility? Maybe it should be, particularly when you're housing animals from the wild that are not domesticated. We've got to interpret the world that they live in, not the world we live in. Can you get oriented in this? You see what you're looking at here? This is a nursery colony of Tadarida, Brazilian free-tailed bats. There's not many in this picture. There is an adult at the very center looking down at you. These animals obviously live in an acoustic world. That's how they visualize their environment, but also an olfactory world. They live in, these caves are nursery environments, and there may be several million bats, several million babies in just a small portion of the cave's ceiling. How many of you have lost your child in a department store at an early age as a toddler, and you go frantically looking for this little person, and you're pulling your hair out? How many of you have done that? Happens daily with these guys. Mom leaves their baby hanging here. They fly off and they forage, and the toddler doesn't stay where they put them. They run off moving around. So mom comes back in the middle of millions of these babies. She's got to find her baby. They don't just nurse another pup. They nurse theirs. How do they find it? Vocalizations and olfaction. That's their world. We've got a raft bat on the left and an elephant on the right. I talked about the ultrasonic vocalizations of the bats. That's how they visualize the environment. They're not seeing what they're not flying into. They're hearing it. They're seeing it through acoustic signature. The elephant, on the other hand, uses infrasound and can communicate extremely long distances with infrasound, as can the cetaceans. So, what sensory modality are we altering when we move into an animal's environment that includes ships out here, that includes sonar that we use? How are we altering the animal's sensory environment? We've got rodents that are herbivores. We've got rodents that are carnivores. We've got carnivores that are carnivores. This is the weasel on the left, one of the world's smallest carnivores. Eat a mouse or two a day. Keep them in captivity. You better have lots of mice, and they prefer them alive. You can train them to dead mice, but that does actually take a little while. Polar bear, on the other hand, you're going to have a lot of different food for them. It's not going to be mice. In terms of what type of research, conservation. Polar bears are having a tough time right now. It appears that global warming is a real thing, and their environment is in trouble. Mammals range from solitary, the lynx on the upper left, to aggregations of thousands. What you can't see on the bottom right is a migration of caribou. All the little spots out there are caribou. So from solitary to very, very large densities, from communal to completely social. You better understand that when you start manipulating the populations, the family groups. Elephants, for example, if we need to reduce elephant populations, you don't take an animal out of a social group, you take a social group out. You've got to understand the social structure. The take home with these, and I think this is true with many of the taxa that we're looking at, is we are working with wild animals. Probably more true in mammals than anything else. We have domesticated many of these animals, many of these species. And what does domestication mean? It means that they need and accept the care of human beings. So we have altered their environment. They respond differently to the sensory cues that we're providing. A wolf will respond very differently to the sound of gunfire than my bird dog does. My bird dog hears the gun go off and, oh, wow, this is good. The wild animals, not so much. When they're not domesticated, when they are truly wild, the biggest stressor in their environment is you. The IACUC is overseeing a situation where the investigator is the biggest stressor these animals face. When we interact with them, we're causing stress. And stress is not a bad thing. It keeps them alive. Distress, on the other hand, is a bad thing. So we want to look at how we're interacting with these animals to minimize stress and to keep it from going into distress to the point that we can. The more we can minimize stress, the more we're going to be able to study the natural behaviors, the natural physiology, the ecology of those animals. And that's what we're focused on. Not using these animals as a model for human systems, but to understand the animal, and that means minimizing our impact. It may mean things because they live in an olfactory environment. It may mean things like limiting the amount of times you touch that animal when you're handling it to put a radio collar on. Minimizing the scent transfer, minimizing the sound that's associated with handling that animal. Minimizing all of those external stressors that they perceive and we don't. I'm going to stop with that. Thank you.
Nope. It's always easier when you're in the audience. All right. We've done land, air, maybe swamp. Let's go underwater, and we're going to do fish. So I'd like to introduce Stephen Cook, who is a Canadian Research Chair in Fish Ecology and Conservation Physiology and professor at Carleton University in Canada. Dr. Cook. It's you.
All right, pleasure to be here. Thank you very much for the invitation. I have the pleasure this morning of sharing with you some information on the diversity of fish. I've been fascinated by fish for many decades, in fact, since I was a little child. What I like about fish is the mystery. You know, you can walk out, you can look into the water, you might be able to see down a few feet at most. It's always uncertain what's out there. And I think it's that mystery which really drew me to the realm of fisheries science. This is the prototypical fish, the rainbow trout, something that I think most of us might think about if I were to say draw a fish on your page. It would generally have this fusiform type shape. This is the white rat of the fish world, at least in North America, quickly being replaced perhaps by the zebrafish. There are other fish that have been largely domesticated, things like the koi, for example, some of the guppies and such. But what I'm going to really focus today on are fish in the wild. So once one sort of opens one's mind and starts to think a little bit more broadly about what is a fish, these are some things that might come to mind. This is a pygmy seahorse, so very different body shape than the rainbow trout in the previous slide. Here's a puffer fish, so an animal that can rapidly increase its size by 2 or 3-fold. Okay. Then we have the good old Greenland shark. So this animal can live over 300 years. Okay, so one of the oldest lived organisms on the planet, spending most of its life under ice. And then we've got the Trinidadian guppies and all their diversity, yet this is a single species. So you're seeing immense variation in color among conspecifics, not just color, other morphological characteristics as well. Here we have a blind cavefish that spends its entire life in darkness. Okay? So when we start thinking about fish and thinking about animal care, we need to remember that not everything is a rainbow trout, not everything eats trout chow, not everything needs 6 milligrams of dissolved oxygen, not everything needs to be held at 18 degrees Celsius in spring water. So that's something that I routinely battle as I work with wild fish. I've got up there the dictionary definition of what is a fish. Ectothermic aquatic chordate, breathing by means of gills throughout life. Sometimes accessory organs are used, such as the swim bladder. And if they have limbs, it's in the form of fins. Oftentimes, when I start off my undergrad class, beginning of the semester, and say, hey, what is a fish? People use words like slimy and scales and so on to describe fish, but that's actually not part of the definition and doesn't necessarily help us to narrow it down to it being a fish. Fish typically live in water, but as a biologist, whenever I get up and start talking, I'm always generalizing, I'm always lying. So there's certainly exceptions, and there's a mud skipper off to the left that likes to perch on rocks. American eel are known to slither around dams by going across wet grass at night, for example. So there's all sorts of interesting adaptations. And then fish are typically ectotherms, although there's some species, such as the opa, that exhibit regional endothermy. So their heart is a couple degrees warmer than ambient water temperature. So fish are certainly the most diverse, numerous of the major vertebrate groups. They dominate the waters, both marine and freshwater, throughout the globe. There's estimated to be around 35,500 species. And currently, if you look in the online database called FishBase, I'll tell you a little bit more about FishBase in a second, there are 33,600 species listed there. That does not necessarily mean all of them have been well described, but that's the current state of knowledge as of today. We're still discovering somewhere around 200 species a year. Some of that is thanks to molecular genetics and simply realizing that these two fish that look an awful lot alike are in fact different, but also a lot of it's simply increased assessment capacity in some of the inland tropical regions that are really quite biodiverse. They're more speciose than any other vertebrate, than all other vertebrate taxa combined. And considering the availability of salt water, most of the globe is covered with salt water. There's more salt water than fresh water out there. Only 58% of the fish are exclusively marine, 41% freshwater, and about 1% move between the two environments. That 1% are some of the most enigmatic and best known species, things like sturgeon and a lot of the salmonids. I mentioned FishBase, so I thought I'd throw this out there simply because it's a good resource. It was created by Daniel Pauly and Rainer Froese, and it is an online community-supported database. It's not owned or operated by any government. It's just based on the goodwill of the community. And so if you've got somebody that's working on fish, it's a good place to direct them. It's got information on some of their environmental and physiological tolerances and information, their feeding ecology and so on. And I think that kind of information is pretty key when you're trying to figure out the appropriate conditions that one needs to think about when interacting with these animals, whether it be bringing them into captivity or holding them temporarily in the field. Fish occupy really diverse environments, temperatures from less than 0 degrees Celsius, upwards of 40, incredible range in pH, salinities from 0 to 90 parts per thousand, dissolved oxygen from 0 to saturation, even supersaturation in some cases, depths ranging from literally at the water surface to the abyss, 7,000 meters. There's been fish documented at that depth. And think about the pressure at that depth. I'll bring up at this point the idea that if you work on fish that are anything below about 3 or 4 meters of depth, you can encounter barotrauma issues as those animals are brought to the surface. There's folks that have come up with creative ways around that. So for example, some folks in Florida actually do surgery underwater when they're implanting telemetry transmitters. So they have scuba divers doing surgery. And then in other cases, there are protocols to bring the fish to the water surface slowly in traps, do your surgery, and then lower them down slowly over a series of days. Obviously, the light levels vary quite a bit depending on the water clarity, on depth, and so on. We certainly have caves that are dark in the northern hemisphere. Well, not just the northern hemisphere, Arctic and Antarctic regions. We have fish that spend their entire lives under ice. And then where I live in eastern Canada, we have a good old winter that's coming shortly, and fish have to deal with things like frazzle ice and anchor ice if you live in a riverine system. So lots of challenges. There's a ton of folks that work in the realm of environmental physiology on fishes. They're probably one of the most studied from that perspective, and that's all really, really useful information for animal care practitioners and scientists to pull upon to understand these various beasts. Fish are also quite diverse in terms of life history. So, for example, lifespans that range from days. 59 days is the current record, if you will, in terms of a fish that completes its entire life cycle. 59 days from birth till death. And it reproduces within that time period, whereas I already mentioned the Greenland shark that live approximately 300 years. There's some thought that it could be over 400, but the techniques used to estimate age get a little bit sketchy that far back. Size range anywhere from a fraction of a gram, so organisms that are about 7 millimeters. You can see that's the smallest fish species. It's the second smallest vertebrate. Apparently there's a frog that's even smaller. Fairly recent find, darn you. But you don't have any frogs that are 34 metric tons. So that would be the whale shark. Certainly diverse breeding strategies, everything from things like scalped hammerhead, which are viviparous and giving birth to live young, internal fertilization, right through to oviviparous. So these are sort of the typical external fertilization where the females deposit eggs and are fertilized by the semen from the male. So think about, for example, a spawning Pacific salmon. That's an example of that strategy. But there's lots of other reproductive strategies. Parental care is reasonably common among some of the bony fish. There are also interesting things that can happen with respect to sex. They aren't necessarily a boy or a girl throughout their entire lives. There can be sequential or serial hermaphrodism, which can further complicate things. Also diverse feeding modes, and this is of course relevant if you're going to be holding these animals for any period of time. It's everything from predators that will require and demand live prey. In the top right, you might look at that and say, whoa, I wouldn't want that to bite me. Well, in fact, that animal's not going to bite you. It's going to crush seeds. So there are fish that focus on things like avocados or nuts and seeds in South America, and that's what that is, a pacu seed-eating fish. Bonefish, bottom left, they basically drive their snout into the benthos and rummage around for various invertebrates. And then to the right here, this is a sea lamprey. In the Great Lakes Basin, they are invasive. And I chair the Sea Lamprey Research Board for the Great Lakes Fishery Commission. So we've got about 200 researchers working the Great Lakes Basin and beyond, trying to figure out how to kill these things, okay? So this is a parasitic beast that attaches to a variety of native fish, and so there's interest in restoring native fish populations to the Great Lakes. This is probably one of the largest and most science-based control programs that exist in the world today, and we spend somewhere in the order of $5 to $10 million a year just on the science side of things and research to support that. And a lot of folks need adult parasitic lamprey to study, and so that means keeping live adult fish that these fish can feed on, and so that creates interesting animal care dilemmas when you know that one fish is essentially being slowly eaten to death by another. So we've actually had a proposal come before us to create an artificial feeding, functionally like a nipple that these things could latch onto and be fed artificial blood, or perhaps blood that's been captured from a cow that's bled out for slaughter. So all sorts of interesting things to think about here. So I've talked largely about species and implying that a rainbow trout is different than a brown trout, which is different than an Atlantic salmon, and that certainly is the case. But I don't want to leave you with the impression that that's where the diversity ends. This is, in fact, an area where I devote a lot of my research to, looking at differences among populations and among individuals. So we certainly know that there's differences among populations in physiology and life history, and I've got some sockeye salmon on the in that image, just to use that as an example. We've done a fair amount of work on sockeye, and they exemplify most of the examples up here. So, for example, there are 120 different populations of sockeye salmon in the Fraser River of British Columbia. And we know that that we have stock-specific thermal tolerances. So if you go to your textbook and say, well, sockeye salmon are X degrees Celsius, we know that you're going to accelerate maturation and kill some and lead to massive early disease development. And in others, you're going to retard their development based on a temperature that you might select. So don't assume that a sockeye is a sockeye is a sockeye. You need to think about, and that goes across all organisms. The more we study, the more we recognize that there's differentiation among populations, and that's something else that's important to consider. We also know that there's differences among individuals. We have bold and shy individuals within populations, and this extends well beyond fish. This is across all of the vertebrate taxa and indeed in invertebrates as well. We see differences in catchability. We've done selection experiments with largemouth bass where we've created fish that are much easier to catch, and we've got other fish that are much more difficult to catch. But that started with natural variation. There's no gene for catchability, but there's a series of correlated traits that make one individual more likely to encounter capture gear than another. Stress responsiveness. We have low responders and high responders in natural populations, and oftentimes we see that being correlated with fitness in both directions, depending on the context. We also see differences between males and females. A couple years ago, we realized, we hadn't been looking for this, but we started to see a pattern. We went back and looked at about 10 or 15 studies that we'd done on Pacific salmon and realized that the females always fared much more poorly, usually 30 to 50% higher levels of mortality in all of the studies that we did. For example, post-capture fisheries interactions or any of the holding studies. The females always died earlier. So the females seem to be much more sensitive, at least within sockeye salmon. And then differences relative to development and nutritional state. So these are all things that one needs to consider. It's not simply thinking that a rainbow trout is a rainbow trout is a rainbow trout. I just want to address the elephant in the room really quickly. This idea of, do fish feel pain or don't fish feel pain? Let's do a show of hands. Why not? Do you think fish feel pain? Hands up. Okay. Do you think that fish don't feel pain? Okay. I would suggest that anybody that put their hand up for any of those scenarios doesn't know what they're talking about. And my reason for saying so is that the evidence base is severely flawed. Okay? I don't know whether fish feel pain or not. I just know that the science that we have to date is extremely problematic. So you can find papers that you will read, peer-reviewed papers, but don't necessarily assume that because it's been published in a peer-reviewed journal that it's indeed of high quality, okay? There's a lot of information that's out there that's not, where the findings don't necessarily jive with the experimental evidence. What you'll see in the fish literature in particular are some pretty extreme, so we'll sort of say wandering definitions of pain that essentially are morphed to fit the experimental evidence that arises from a given study. So we've gone through and we've got a paper that we published called Do Fish Really Feel Pain, where we evaluated some of those claims, and with a group of 12 other co-authors representing folks with expertise in pharmacology, in neuroscience, and so on, we've, in our minds at least, put some serious question into whether or not they do or don't. And I do a talk on, do fish or don't fish feel pain? And I start out and I go through using the media to help me. And I basically got about 20 slides, and I alternate at about 3-month intervals going between fish do feel pain according to scientists, and then and to the right, anglers are finally off the hook, fish feel no pain. Basically, every time there's a new paper out on either side, this gets splashed about in the media. So be very careful with what you read. Use your critical thinking ability. This has implications for how we operate, the use of drugs, okay? Putting analgesics in fish in the wild. I would submit that that is largely illegal. You're putting opioids in fish that could enter the human food system. I have to routinely tell my veterinarian that that is illegal, that it's against Health Canada regulations, and I assume it would be against regulations down here too. To juice a walleye up with opiates that's going to be caught the next day and consumed, that's problematic. So be very careful when operating in that realm. My take on what we need to do a systematic review, bringing all folks to the table and try and figure out what's going on. That has yet to be done. Right now, there's 2 very different camps out there, and they're making it very difficult for animal care committees to know how to operate in this sphere. In terms of the fish study questions that we pursue, there's certainly many fundamental questions, and these transcend taxa, the hows, the whys, a lot of effort trying to understand fitness correlates, understanding evolutionary relationships, and so on, and certainly understanding how fish make a living in very heterogeneous and dynamic environments. The applied questions, this is certainly where more and more folks are at least getting the money to do the research and spending their time. This is also where the natural resource management agencies play. So addressing things like hydropower interactions, connectivity questions, entrainment, that's things being sucked into other things, or GBT, gas bubble trauma. There's probably more money applied research spent on fish as it relates to interactions with hydropower than any other conservation or resource management issue in North America. The Columbia Basin alone, it's many, many billions of dollars spent on an annual basis. Fisheries planning and management, things such as determining where protected areas should be placed and do they work. Quantifying and reducing bycatch and release mortality from fisheries. looking at habitat enhancement, stocking activities, and evaluating fish responses to various stressors, everything from noise to light to chemical and so on, and certainly the various climate change-related stressors, and then identifying critical habitats. I've got a caveat here, or a note, and that's that applied fish research almost always means engaging with stakeholders, okay? So this is a little bit different than some of the other taxa that we've talked about. Yes, somebody looking through binoculars at birds, there's a lot of citizen science programs that incorporate those approaches. But sometimes we actually recruit commercial fishers to fish on our behalf. We'll go and work alongside anglers that are actually doing the capture component of the interaction. We may, in fact, hand them the tagging gun and say, can you put an anchor tag in these animals? We may leave them with that tagging gun to continue that research after we disappear. So it also means perhaps working on board a commercial fishing vessel. And I can assure you, the most rapid way to lose your credibility is to start pithing every single fish that you interact with, okay? When they're harvesting 100,000 fish and putting them in the hold and not pithing them. So there's a lot of interesting things that come up when it comes to dealing with stakeholders, things that might require one to modify one's behavior and work slowly to educate instead of going in and saying, nope, this is how we do things. It's a two-way street. You're there to listen and learn as much as you're there to teach. Approaches to studying wild fish in the lab. So this is where we go out. So that's one of my students, Clay, in the top corner. He's gone out and netted lionfish off of reefs in the Bahamas. He's brought them back. He's going to hold them in captivity for about a week or two, and then those fish will be euthanized. They're an invasive species, so we're not going to return them back to the environment. And then the bottom, we've got folks that bring animals in and essentially try to domesticate them. So things like guppies, or the Trinidadian guppies, where there's a number of colonies that have now been established in North America, and researchers trade guppies back and forth to increase genetic diversity. But those animals are not going back to the wild. And then we've got various salmonids that we can bring into the lab as well. What you can see here, you can see sunlight in the background. This is at a Fisheries and Oceans Canada facility. A lot of the labs that I'm talking about when we talk about working with wild animals are things that have that are outdoors or based at field stations and such. They tend not to necessarily be on university campuses in the basement of buildings. This is more of my realm. I tend to do things sort of in mesocosms or the true field. There are groups like the Illinois Natural History Survey that have experimental pond facilities where one can essentially set up mini ecosystems and stock the ponds with what you want. There are net pen enclosures that can be set up. This is the Shark Lab in Bimini working on lemon sharks, and you've got an observation platform above. You've got various mesocosms here. These are for combined fish zooplankton studies. And then experimental channels. This is at the Oregon Hatchery Research Center, operated by Oregon State University. So very common. When we're in the field, the true field, we can do work that's observational. So this is not touching fish. One can use a variety of imaging technology. This happens to be DIDSON, so we're essentially taking underwater images using hydroacoustics. We can do video. This one is baited, so certainly modifying the behavior of those animals, putting bait out and drawing them in to see who's around and who's interested. And then here we've got snorkelers with a consulting firm, and they're swimming in a stream and they're counting fish. So that's very common for things like salmonids. Obviously, you would move fish. You would alter the behavior of fish with something like that. Oftentimes, we need to capture fish. We need to handle them. And so the variety of tools that one can use, and I can just give you a brief primer here. In freshwater, a common one is electrofishing. Very effective in small streams, lakes, reservoirs, rivers for accessing fish. This is Neil Hammerschlag's group from the University of Miami, and they use drum lines to catch a variety of large shark species. At the bottom here, this is a trap, so the tide is out. So this is in the Minas Basin, in the Bay of Fundy. in eastern Canada. And what happens when the water levels are up, the sturgeon swim in, the Atlantic sturgeon, and when the water recedes, they're stuck within this trap. So this is a historic fishing method, and it's now what the scientists use to access fish, working alongside the commercial fishery. And then this is an example. This is Alice, one of my grad students, and we were working at a fishing tournament a few weeks ago. So we weren't handling the fish until they were basically delivered to us after they'd been weighed in. So exploiting existing capture platforms. And then are the interventions that occur, and they can be everything from surgical. So this is in the Bahamas. I'm implanting a transmitter in a bonefish. Down here, we've got a pop-up satellite tag on a billfish, a jaw tag. There are thousands, 20, 30, 40,000 of those put out in the Great Lakes each year to identify walleye for mark recapture studies. Here's one of my ex-postdocs, Austin Gallagher, bleeding a shark, so obtaining a blood sample from a shark that will be released shortly thereafter. There's been a lot of innovations in what we'll call field physiology, sort of the field physiology toolbox. We can now use point-of-care devices on boats. We can tag animals with various biologging and transmitting devices. And so you've seen probably on your animal care committees, I would imagine, a lot of increase in the last few years of folks interested and able to work on wild fish. Bringing wild fish into captivity always presents some challenges. What I tend to think about is sort of the short-term thing. I'm not talking about trying to domesticate these animals more permanently, but short-term for experimentation or evaluation. Fish always have to be captured, and it doesn't matter what gear type you use. Fishing, the capture devices, capture approaches we have available will always cause injury and will always cause stress. You cannot catch a fish without causing some level of stress or injury. The injury could be very minor, just dermal abrasion, a little bit of fin fraying and so on, or it could be quite extreme to the point where the animal is dead at the point of capture. Same with stress. That animal is going to be removed from its natural environment. It's going to interact with humans. It's going to be placed in a different environment. There will be a traditional glucocorticoid stress response. It's impossible to avoid that. Wild fish then have to be transported to the lab facility. That's inherently challenging. Michael Murray from the Monterey Bay Aquarium is here, and I'm sure he can tell you all the challenges that they face with moving some of the larger beasts, things like tuna, for example, which are not easily moved. Ram ventilators, very difficult. Essentially have to shove water in their mouth continually. Then wild fish like wild food. You cannot take a largemouth bass that's 16 years old and is eating frogs and, frogs, crayfish, and other bony fish for its entire life and think that you can put it on trout chow and that it's going to do well, okay? Wild fish carry a variety of pathogens. Indeed, I would challenge you to find a pathogen-free fish in the wild, yet whenever we bring them into captivity, the first thing we want to do is nuke them with antibiotics. It doesn't make a whole lot of sense, okay? Similarly, with tagging, a lot of people say, oh, well, we've just inserted an electronic tag and we're going to let that animal go. We should give it a hit of antibiotics. Dan Mulcahy, who couldn't be here, has written about the problems of doing that, and that's something that I always have to fight against with my animal care protocols. I don't know what that's going to do to the natural flora that's above that exists within that organism. We're increasingly learning about the gut microbiome and its importance in a variety of ways in mediating the health of fish. And not necessarily because an animal has a pathogen aboard is it necessarily in a disease state. So it's only when animals tend to be compromised, perhaps because of lack of appropriate food or some external injury, that we see things move from being pathogenic to more to the animal being truly in a disease state. And then also, wild fish vary in their health and condition, okay? When you're out there, if I get a fish and it's got a big lesion on its side, should I high grade and avoid tagging it, or should I tag it and realize that that animal's part of that's just what that animal had? Or should I euthanize it because I'm supposed to euthanize animals that are injured or stressed according to a given protocol? So these are things that one needs to think about. not all fish are in pristine condition when you interact with them in the wild. I'm going to end with this slide on resources. There aren't a ton, and most of them that exist are out of date. That's one of the biggest problems. Because a lot more people are working on wild fish in the wild, it's very much a frontier research area. We're pulling upon research that's been done on other taxa in many cases, for example, ectothermic work in particular that's been done on things like turtles, basking turtles, and trying to apply those same techniques to fish. There are guidelines for the use of fishes in research that were developed by a number of US-based professional fisheries organizations, but they're already out of date as well. 2014 is the most recent update on those. And then if you want to read something where I whine about some of the realities that I've faced over the years, this was developed with with authors from 4 different countries, so this is not just a Canadian perspective. And in here we hit on 10 things that we want animal care committees to think about when they're evaluating protocols. And one of them I touched on already, that idea that don't overprescribe, whether it be pain meds that are illegal or we don't understand how they work, whether it be antibiotics, and even thinking about when it makes sense to use anesthetics in the wild. Of late, we've been using electricity, electroanesthesia or electrosedation. It works really, really well. You don't have to deal with any of the issues with the animal having a drug burden, and the moment you take your hands off that fish, it swims away. So there's gloves that we wear. The moment you take your hands off the fish, they swim away. So your pre-op and post-op care is much, much easier. But at this point, we have very little information to go on from an animal care perspective, and only 3 published papers in the literature. So animal care committees are having a real tough time wrestling with that, trying to figure out whether or not that's something that they should be endorsing or running from. Thank you.
All right, I'd like to call up our speakers if there's questions. If there's questions. Oh, we have one in the back. We use microphones.
Hi. So I think I have a question for Dr. Cook, who just spoke. So I guess maybe I misunderstood what you were saying about fish in pain. And so in terms of the IACOX, and we've been talking about animal welfare, I guess it's surprising that we question a vertebrate animal maybe not feeling pain. I don't know what kind of studies are really necessary to think they might not. And I guess in terms of the IACOX, like, how do you take that into context in determining animal welfare? Because if we're now questioning whether fish feel pain and need certain studies to really think that they may not or may, or I don't know, this objectivity, then how do you bring that back to your IACOC in terms of telling people how they should appropriately treat fish in the wild?
Right. So animal welfare can be considered independent of whether or not an organism feels pain or has the capacity to suffer in that way. And so I'm certainly pro-welfare. So for me, what we do, aside from applying analgesics, is no different. So for me, the issue is analgesia and random application, especially in wild animals, not knowing what it does to the physiology of an organism. We barely understand how anesthesia works in humans. Seriously, talk to an anesthesiologist and ask them about their practice and the detailed mechanistic basis for how that functions. We simply have no clue how that plays out and works in fish. So I like to follow the evidence base, and we don't have an appropriate evidence base to guide us in the application of those pharmacological products in terms of giving them to fish, whether they be captive fish or wild fish. Right. So anesthesia, I think of in fish for sedating the animal so we can do the procedures that we need to do. Whether or not that's for pain management or not, I'm unsure, but that should not be coupled with the analgesia. So the analgesia is the particularly problematic part. We use anesthesia all the time. I use MS-222, I use Clovoil, I use electroanesthesia, but there are other instances where we don't. Sometimes you're tagging fish. I've got colleagues that do work in the Arctic, do the tagging of the Greenland sharks, do tagging of Greenland halibut. If you use a traditional chemical anesthetic on those animals, you have to hold them for 2 or 3 days before they literally are able to move again. So there's a again, that's the physiology of the animal needs to be the driver. When we talk about physiology, we can talk about facts and function, and that can be done, in my mind, independent of any discussion of pain. And welfare can be brought about with an understanding of the physiological functioning of an organism.
My question is on herps and reptiles in particular, the wild captured reptiles. You made a reference to checking them sometimes every other day. And I had an investigator who wanted to do that, and PHS gave me a ration of, we expect far more than that, and that is entirely unacceptable, and on and on and on, despite the fact that some of these were wild caught individuals. And I'm like, yes, sir, we will look at them daily. Some of them were domestic, but how did you deal with that from a compliance standpoint?
I haven't been questioned a lot on that, but what we tend to do is the IACUC will write a deviation, and we'll write a paragraph up on why, how, and what the scientific rationale is, and the IACUC votes on that and keeps that in their policies and procedures. And when questions are asked, here's what our IACUC believes is the best for animal welfare for this specific group of taxa.
Okay. Were they PHS assured? Those
Yes,
animals?
we are. Yes, they were.
Okay. Thank you. I appreciate that.
If I could tag in on that, we had an investigator working with amphibian salamanders, and they were going through winter hibernation brumation in a substrate, so they were 10 or 12 inches under the surface of the substrate. To check that animal, you'd have to literally excavate them every day. You can't do that. And they are in brumation. The same thing occurs with bats. If you check a bat every day by opening a box and looking at the bat to make sure it's alive, it won't be alive very long. They're using up the energy stores to arouse because you're disturbing them. Either we need to come up with a different way of evaluating that, taking into account that they're hearing frequencies of 100 kilohertz or something like that, or we need to work with the physiology of the animal. And what the approach we took is exactly what Chris laid out, is to write a deviation, have the IACUC review that and approve it, and that's entered in the record.
I was pleased to hear you talk about the avoiding antibiotics in fish for many reasons, upsetting the gut biome or the skin biome and other things. I'm curious whether any of the other taxa are also looking at that issue, and are we potentially overusing prophylactic antibiotics?
To be honest, I'd have to pass that one off to Tracy. But from committees that I have sat on and veterinarians that I've worked with when we're implanting telemetry devices and things historically, we haven't had that issue. But that's just one little program.
I have a question in terms of live food sources. So in general, if you need a live animal to feed your animal because it won't take the dead one, and the committee comes back to you with comments and concerns, basically I wanted to know what additional information have you provided to them to basically, my issue is the distress it causes the rodent, and in terms of the awareness of that in comparison to some of the other species that are used in the work that is done. So I just wanted to get some commentary in general of what you have provided and experienced in that dialogue.
So we work very hard to use dead animals. So, and you can, with most snakes, you can get them over to eating dead. But sometimes, no, you can't. And our vet and our committee honestly hasn't questioned at that institution, questioned that aspect. We do everything in our power to use frozen, thawed animals, warm them up, and there's a lot of tricks you can do with the animal that's deceased to do that. But my veterinarian at that time basically said the welfare of this animal is paramount, and we want them to feed. I give you permission to utilize, a culled live mouse for that.
So the fact that it's an endangered species is not an animal welfare issue, except for the permits question. That would be true for any owl or any raptor that goes after live prey, regardless of its protected status. And we're talking largely the United States here in Canada. When we're talking about those kinds of permit issues, other countries do have permit issues, but in the context of this conference, mostly United States and Canada. So I encourage you to not think of the permit issue when it comes to animal welfare and how you're going to keep the animal alive. So the context of using live bait also comes up in a different way that you might not have thought of, and that is using live lures to capture the animal, because obviously it is bait. In that case, it's done in a double cage where the animal is protected. And there are standards for how often to swap out the animal. But in terms of, and providing food and shelter and so on while it is in the cage. But in terms of providing food, there are in fact like little robo devices that you can use that you can pull it on a string. You can do lots of things to get it to feed. It's if it works, great, but if it doesn't, I concur that the issue is to get the animal to feed. And so try it. If it works, great. If not, then you've got to go back to using live rodents.
I just have I just have a little, I guess it's more of an advertisement to the previous comment about microbiome changes in response to any of the treatments that we do. As part of my clinical role, I work with the reptiles and amphibians and fish, but in my research, I actually look at environmental effects of how we're housing animals on the microbiota and how that changes the animal models. And so with that, I also work with the Notobiotic Group. I'm co-director of our national center and do a lot with the microbiome. So if anybody is interested, right now I'm working on the effect of just daily treatments on our mice that we have housed. But if anybody's interested in chatting about maybe doing collaborations with the other species, I'd also be interested in that, because I do a lot of work with the amphibians as well. So
Let's
come
chat
find
in the
me.
hallway, because I know people working on that for birds.
And birds. Okay. Awesome. Cool. We don't have birds, so you guys are on your own. But
Along those same lines, it's maybe a little far afield for this group, but there is a huge interest and concern about antimicrobial resistance out there in the world these days. And so giving single shots of antibiotics and sending them back out into the world is not a good idea.
So I have a question actually mostly for the audience, less for our panelists. We talk about these live lure animals or these bait animals, not just for food in captivity, but that's inclusive. So I'm kind of curious, when you're reviewing as an IACUC, that you realize that live bait or live lures or live prey needs to keep your target animal alive or to capture it, how are you reviewing that in your IACUC protocols? And if it is reportable to USDA, what category are you putting it in? Any thoughts on that? So just wanted you to think about that.
In Canada, we list that as a category D, and we have the PI come to talk about that with our committee. It's a predator-prey situation, which takes a lot of further thought on our part.
Patty, we actually had this come up in real life last year, and I gave my opinion, but I'm not the judge, so I suggested they contact animal care, which they did, and the answer was not covered. And my suggestion was use a purpose-bred mouse of the genus Mus, and that way we don't even have to have the discussion. But even if you did want to use a non-exempt animal, the answer from animal care was not covered.
So that's interesting. So usually we wanted to know whether that was a category E, since category D would require some type of anesthetic or anesthesia. Category E would be not using that, and it would be under a certain type of distress. So that would be another question for any of our animal care folks who might be with us.
I'll offer
I know, I
one.
didn't want to put you under the bus on that one.
I'll offer one scenario that we face at my institution with live prey. This was on reptiles. We had newborn water snakes in captivity. The focus was looking at growth rates of these snakes, and the student working with them was feeding them killed fish. But he found out that some of the snakes were not feeding on the killed fish, and we took that step to minimize pain and distress to the prey, obviously. But since they weren't eating the killed fish, we were causing pain and distress to the target animals. And what we wound up doing is switching to live prey, live fish, purchased at a bait shop, brought in that day, fed, and eliminated, not covered.
If I can jump in regarding regulated species of animals that are used for bait or food. I have not had this question come up in a long time, but the last time it did, checking with leadership, food, fur, or fiber. Animals used for food, fur, or fiber are not covered. However, that does not mean it's not a welfare issue. It just means that it's not covered.
Hi, I'm that chucklehead fish head from Monterey, and we look at things a little bit differently in the public aquarium world. While we do have an IACUC and we are a licensed research facility, we're also exquisitely transparent, and we spend a lot of time taking the pulse not only of our visitors that come through, but also the general public, the people who don't want to come to a zoo or an aquarium, and what they're thinking. And I think as a scientific group and as an animal welfare group, we need to recognize that the importance of animal welfare is increasing dramatically within the public world, and that includes fish. That includes invertebrates, and we need to start recognize that as we go through this IACUC process. Those are the folks that we're going to be asking to fund our research in the future as, federal funding starts to evaporate. Those are the folks that are going to listen to the scientists. So we've got to remember that. And while Dr. Cook is very, very correct in we don't know about fish pain, the public in many, many circumstances actually thinks animal welfare for fish, for jellyfish, is very important, and we can't ignore that fact. Now I'll get off my soapbox and go back into my fish tank.
Our last question.
Thank you. Bob, you mentioned that there's certain small rodents, when wild caught and placed on like some of the bedding, turn into gooey, sticky balls of fluff. What substrate did work for those? I couldn't quite catch that.
It really depends on the species as far as the substrate. The grasshopper mice, we kept them on some stuff called PJ Murphy hardwood bedding, but it's their finest grade.
Oh, okay.
You can go to sand, and sand works great for them.
I was wondering.
It's just really, really heavy and difficult to deal with.
Yes.
But the key point is matching the substrate. Just kind of to follow up on that, if you're working with something like voles, they not only need an appropriate substrate, they also need a cover over the top. So hay or something that's just thrown in there loosely that they can make tunnels underneath, it's about the only way you're going to get them to breed consistently, at least some species.
I have a feeling we may be bringing in Allegheny wood rats.
With wood rats,
Allegheny wood rats.
the wood rats I've kept, actually I kept on more like Santa chips and what have you. But the key with wood rats, at least what I found, is giving them something to hoard. So I would keep them in cages, and instead of using food in the hopper, I'd throw the food loose in the cage. They would hoard the food. I was also cutting up strips of cardboard or put in sticks, let them build a nest in one corner.
Okay, so they need a lot of enrichment and that kind of thing.
Yes.
Thank you very much. I appreciate it.
Let's give a round of applause for our speaker. Oh, yeah. And a final statement, perhaps?
Yeah, I've heard the word evidence being used a lot over the last few days, and I know we all throw it around, and oh, here's one paper that says this or one paper that says that, and we got expert opinion here or there. Think about the medical realm. You may be familiar with something called Cochrane reviews. So when you go to a physician and they decide whether or not you're going to have a pap smear this month, or whether or not they're going to give you an antibiotics for the common cold, that's not them shooting from the hip. What that is rigorous evidence synthesis process that's repeatable, that's transparent, that evaluates the quality of the evidence that's brought to bear on a specific issue or topic. Unfortunately, given the diversity of species we're dealing with and the diffuse literature base on frontier topics, we can't do that. So I think that I think that's something that we should aspire to do true evidence-based conservation. Those that are veterinary practitioners, it's all about doing good and not doing harm. But at the end of the day, we do not have a high quality evidence base to draw upon. You would not want physicians making the kind of decisions that we're making for your children or for your loved ones. We just don't have that same quality of evidence. So just think about that and be very critical of all of the pieces of science that come across your desk. As an editor, as an author, as a peer reviewer, I can tell you that you can pretty much get anything published. And just because it's got a peer-reviewed stamp of approval doesn't mean that you should not pull out your bullshit meter and give everything a really, really critical appraisal when it comes across your desk.
Thank you.